So my presentation is called Detection of Rh Antibodies in Patient Plasma using Customized iPSC-derived reagent red cells. So we'll start with a little bit about sickle cell disease and transfusion therapy. Sickle cell disease is an inherited red cell disorder where the patient's red cells make hemoglobin S, and it causes the cells to sickle under hypoxia. It affects approximately 100,000 people in the US, predominantly who are of African ancestry, and red cell transfusions are currently used as one of the primary treatments for patients with sickle cell disease. But alloimmunization to the transfused red cells still remains a major complication. In fact, 30 to 50% of patients who are chronically transfused are alloimmunized. And that's partially because you just can't match for all the antigens that are on the red cell surface. There's a ton of stuff on there. So there are 352 red cell antigens encoded by 45 genes and about 2,000 different alleles on the red cell surface. Fortunately, not all of them are equally immunogenic, so you can prioritize what to match for. So patients who don't have sickle cell disease but need transfusions are matched for ABO and RHD, which are the most immunogenic, but patients with sickle cell disease and thalassemia are matched for additional red cell antigens, RHC, E, and KEL. But despite this extra serologic Rh matching strategy, these patients still become alloimmunized against the Rh system. And you can see this in the 2013 paper that Stella and Connie wrote, where 2/3 of the antibodies that these patients made are still directed against the Rh system. So the Rh system is not only the second most immunogenic after ABO, it's also highly polymorphic. It's only comprised of 2 genes, RHD and RHCE, and because they're a duplicated gene family, they're 97% identical in sequence. So the 5 conventional Rh epitopes are RHD, which is encoded by the RHD gene, and big C, little c, big E, and little e. And the C's and the E's are encoded by the RHCE gene. And while you can or cannot have RHD, you have to have one of these 4 combinations of RHCE, little c, little e, little c, big C, little e, or big C, big E. And these are the 5 conventional Rh antigens. But there are actually a lot more variation in the Rh system, and about 700 different RHD variants and 150 different RHCE variants have been reported. And these variants are much more common in patients with sickle cell disease. The 2013 paper and the 2018 paper by Stella and Connie also found that 1/3 of the RHD alleles and 3/4 of the RHCE alleles had been altered in patients with sickle cell disease. And this ratio was similar in African-American blood donors. And if you look at the individual level, it meant that about 85% of these patients had at least one Rh variant allele. So this variation is really significant in our patient population. And what's important to note about these variants is that serologic methods that are currently used can't tell whether these variants are present. It can really only type the 5 conventional Rh antigens. So these variants are really important to be able to parse because it can lead to alloimmunization. And one example of this Rh variant is the DAO4 RHD variant, where you just have these 2 SNPs on exon 5 and exon 8, and it causes the patient's own red cells to be lacking certain RHD epitopes that would be found on conventional RHD. So this patient, if they had this variant, would be D positive, but if they were transfused with another unit that was D positive but had a conventional RHD allele, they could be at risk for making an anti-D, even though they themselves are D positive and got D positive blood. So conversely, Rh variants can express these rare or new Rh epitopes. And the example here is the little c, little e, CE733G variant on the RHCE allele, where you just have this one SNP and it causes the expression of the rare VVS epitope that's just not found on conventional little c, little e alleles. So if both the C733G red cells and a conventional little c, little e red cell would both type as little c, little e. But if you accidentally give this to a patient who's only little c, little e positive without having this variant, they could be at risk for making an anti-V or an anti-VS. So having these alloantibodies can be really dangerous, especially if they're re-exposed to the same antigen again. So prior to each transfusion, patients are screened for unexpected antibodies. And the way you do that is by taking their plasma, which may or may not contain antibodies, and then incubate them with reagent red cells with predetermined combinations of red cell antigens. And you're really looking for both a positive agglutination reaction, so the clumping of these red cells caused by the antibodies in the patient plasma, along with negative agglutination reactions where there's no agglutination. And this agglutination is graded and detected on a gel card assay. This is a standard assay that's used in blood banks, where agglutination is graded based on its strength and its ability to pass through a gel matrix that's at the bottom of each column. So if there's no agglutination present, the red cells can freely travel to the bottom of the gel matrix, whereas if there is any sort of agglutination, then depending on the strength, it can get totally stuck up at the top of the gel matrix or pass through, making this cloudy gradient. In patients with sickle cell disease, some antibodies can be really hard to pin down, which is why we want to make these rare IRBCs. So in this first scenario, you could have a patient who is little e positive, but made an antibody that looks like an anti-little e. And the challenge in the blood bank is that you don't have reagents to be able to tell the fine specificity of these Rh antibodies, because anti-little e and anti-HRS, which is a high prevalence Rh antigen, and anti-HRB, which is another high prevalence Rh antigen, all look very similar when you look at it with screening cells. So the IRBC solution is that we can make IRBCs that are negative for each high prevalence antigen each, that are both anti-little e positive, that would let you parse if there's an antibody against a high prevalence antigen or not. And in the second scenario, you could have a patient whose plasma didn't show any antibodies during screening, but cross-match finds that one of the units is incompatible. And in this case, it suggests that you could have an antibody against a low prevalence Rh antigen. And the current challenge in the blood bank, again, is that you just don't have the reagents to be able to say for sure that there's an antibody against the low prevalence antigen, such as GOA and DAC. And what we want to do with IRBCs is to generate red cells that are positive only for this one specific low prevalence antigen, so that you would be able to tell for sure what the antibody specificity is. So we have two approaches to doing this. First is to reprogram rare donor cells, and we have 3 lines that we generated using this approach. The first 2 are lines that lack high prevalence Rh antigens. So these are antigens that are found in 99% of Rh proteins. And the line that we have is negative for the high prevalence antigen HRB, while positive for the other high prevalence antigen HRS. And the second line that we have is the reverse of that. So negative for the high prevalence antigen HRS, positive for the other high prevalence antigen HRB. And then our third line expresses a low prevalence Rh antigen V and VS. And then our second approach into making these rare red cells is to use CRISPR-Cas9 and other zinc finger targeting to gene edit existing iPSC lines. And we used this approach to make lines that we totally couldn't find donors for because they're so rare. And the first line that we made is the Rh null line. So these are red cells that wouldn't have any Rh on the cell surface. And then using the RH null line as a background, we reintroduced RHD to make a line that we call D-, which is a line that has the RHD protein but lacks RHCE. Again, both of these are extremely rare, so you couldn't really find donors for. And then using a similar approach to making the D-, we made variant RH lines by reintroducing these variants in the RH null background. And we made the DAC positive and the GOA positive line, both of which are RHD variants. So this is our schematic to generating the RH null line. We started out with a D- iPSC line so that we didn't have to worry about D at all. And then we knocked out RHCE using two guides, one in intron 1 and one in exon 2. We made a 1 kb deletion that we screened by PCR. And when we differentiated our iPSCs into red cells, we saw that RH expression was knocked out from the cell surface in the RH null IRBC, whereas in the untargeted IRBC, we do see RH on the cell surface. And then using the RH null iPSCs as a background, we reintroduced RHD into the AAVS1 safe harbor locus. We screened using PCR again, and then when we differentiated our cells and then looked for RH on the cell surface, we saw that RH expression was restored, whereas the RH null cells that were used as the parent did not have any RH on the cell surface. It takes about 20 days to go from iPSCs to mature IRBCs. We start out with iPSCs and we do 8 days of EB differentiation where we collect progenitor cells that bud out, which are mostly 235-41 double positive. And then after 12 days of erythroid differentiation, we get really nice orthochromatic erythroblasts that are mostly, and we get a fairly pure population of these cells, 235-71 double positive and band 3 positive, alpha integrin 4 or CD49D negative. All of our cells, for the most part, stay nucleated, but the nucleus gets really compact, and most of them are eccentric, but they do remain nucleated. These IRBCs also express a combination of embryonic and fetal globins. So this is HPLC that Malik had run for us, and you can see alpha chains and gamma chains along with epsilon and zeta. We don't see any beta. And if you look at tetramers, we end up seeing about 25% of our tetramers being hemoglobin F. And maybe more exciting, maybe not, our IRBCs express predicted RH antigen phenotypes. So if you look at the untargeted parent IRBC that we use to make the RH null line, you can see agglutination against anti-little c and anti-little e. And this typing was done using monoclonal typing reagents. And you can see this nice expression over here, whereas in the RH null IRBC, we don't see any agglutination against any of the 5 conventional RH antibodies. And then again, with the D-, we see positive typing with anti-D monoclonal typing reagent, but no agglutination with any of the anti-RHCE antibodies, exactly as we would expect. And then similarly with the GOA positive and the DAC positive, because they're RHD variants, we only see anti-D typing and no agglutination against any of the RHCE antigens. So going from using monoclonal typing reagents, which have a really high titer, and moving towards having IRBCs be able to detect lower titer antibodies in patient plasma, we had to use Physyn treatment with our IRBCs. And Physyn treatment is just a standard thing that blood banks and reference labs do to enhance RH reactivity. And we had to do this because we just weren't seeing such a robust agglutination pattern with IRBCs. So as an example, this is a card with untreated IRBCs. We always start with positive and negative donor red cell controls, and followed by a panel of IRBCs. And this whole card was incubated with patient plasma containing anti-big C. We see this really nice agglutination pattern with the donor positive control, no agglutination with the donor negative control, and then moving on to our IRBCs, we don't see any agglutination with the RH null line as we would expect. We don't see any agglutination with a D minus IRBC, again, as we would expect. And then we don't see any agglutination with the big C negative IRBCs, as we would expect. The problem here was that with our big C positive IRBCs, we would want to see a really strong agglutination pattern, but what we were seeing was fairly weak. It was hazy, and we wanted to see if Physin treatment would make this a little more robust. And that's exactly what it did. Physin treatment didn't interfere, didn't make our cells any stickier, so we don't see any unwanted agglutination with the RH null or the D minus or the big C negative IRBCs, but we see a nice robust agglutination pattern with our big C positive IRBCs. What was also nice about Physin treatment is that it eliminates other blood group antigen expression from the cell surface. So without treatment, we were seeing some expression of the MNS system, so the glycophorin A's and the glycophorin B's. We were seeing the KEL system expressed and just a little bit of the Duffys on the IRBC surface. But with Physin treatment, we basically eliminated the MNS expression from the red cell surface, so it meant that the epitope was no longer there. The KEL stayed as we would expect, but the Duffys cleared up a little bit also. And what's nice about this is that it allows us to focus on the RH and KEL systems, which we already know are a little more pertinent for our patient population. So this is our first line to show that IRBCs are able to detect anti-D in patient plasma. Same format as previously. We always start out with donor positive control and negative control, where we see expected agglutination patterns. Anti-D in patient plasma didn't agglutinate with RH null cells, suggesting that the antibody that's in the plasma is somewhere against the RH system. We see this nice agglutination pattern with a D minus IRBC, suggesting that the antibody is again somewhere on RHD instead of RHCE. We don't see any agglutination with a D negative IRBC, and we see this really strong agglutination pattern with a D positive IRBC. So having the D minus line is really nice because it tells us immediately that the antibody is somewhere against RHD. Well, going further, we wanted to see if IRBCs could detect antibodies against RHCE in patient plasma as well. We saw this card previously, but basically it shows that we were able to detect anti-big C in patient plasma because it only reacted with IRBCs that were big C positive and not with the RH null, not with a D minus, and not with a big C negative IRBC. And similarly for patient plasma containing anti-little c, we only see agglutination with the IRBC that is little c positive and none with any of the others. Again, the same with big E. We don't see any unwanted agglutination, and we see a really nice strong agglutination pattern with a big E positive IRBC. And with an anti-little e patient plasma, we don't see agglutination with the RH null, D minus, or little e negative IRBCs, but we do see agglutination with a little e positive IRBC. So what we really want to sell the IRBCs on is that they can detect antibodies against high prevalence RH antigens because they can often look like something else. And in this example, if you incubated this patient plasma with these 3 screening cells, which are always the same, the first being a D positive, big C, little e, homozygous donor cell. the second being a D positive little c big E homozygous donor cell, and a third that's D negative and little c little e homozygous, you basically look for the common denominator in the agglutinated cells, and you would think that it looks like an anti-little e. But if you look further with the customized panel of IRBCs, you would actually realize that it's an anti-HRS, and that's because you don't see any agglutination with the RH null, suggesting that the antibody is somewhere against the RH system. No agglutination with the D minus minus, suggesting that it's somewhere on the RHCE system and not RHD. And then you have our two IRBCs that are both little e positive, but negative for one high prevalence antigen each. So the first one is HRS negative, HRB positive, and the second one is HRS positive and HRB negative. So looking at these two agglutination patterns, you would actually say that this antibody is anti-HRS and not just an anti-little e. And then here we also show that IRBCs can detect antibodies against low prevalence Rh antigens. So in this case, when you incubate this patient plasma with these three screening cells that have the same antigen phenotypes as the previous slide, you would think maybe there's no antibody in the patient plasma. But when you do the cross match, it comes out that one of the units is positive, which makes you think that maybe there's an antibody against a low prevalence Rh antigen. So you would move on to our customized IRBC panel for low prevalence Rh antigens. And if you see this really strong agglutination pattern with an IRBC that's GOA positive, but no agglutination with IRBCs that are DAC single positive or VVS single positive, it would lead you to conclude that the antibody that's in the plasma is against the low prevalence antigen GOA. So this is a quick summary of how we think IRBCs can facilitate antibody identification in patients with sickle cell disease. So if you see a positive antibody evaluation, you would go on to use our IRBC panel for screening antibodies against high prevalence antigens, which would allow you to quickly determine whether the antibody in the patient plasma is actually against a high prevalence antigen or not. And then in our second example, if you don't see any antibody in the evaluation, but see a positive cross-match, you would go on to use our IRBC panel for low prevalence Rh antigens and be able to tell whether the antibody is against a low prevalence Rh antigen or not. So IRBCs have so much more potential, even though it's cool that you can use it as reagent red cells. And like Stella mentioned earlier, especially Rh null IRBCs, they have a huge potential in being used as other things. And one of the things is to be used as drug carriers. And the Rh null cells are especially important because they don't have any Rh protein on the cell surface, meaning that they're less immunogenic. But for these cells to be used as drug carriers, they have to fit into capillaries and they have to be able to bend. So in our preliminary evaluation of the deformability, we saw that lacking the Rh null protein doesn't seem to make the IRBCs more or less rigid. Even though this does have a caveat in that we're working with nucleated cells that are already fairly rigid, the difference in deformability between the wild type and the Rh null IRBC was not significant. And maybe way down the line, if we can make enough of these red cells to be able to transfuse, Rh null cells, IRBCs have a huge potential in that, again, they would ideally be less immunogenic because they lack Rh protein from the cell surface. But if they're going to be used as transfusible blood products, we need to show that they have the same oxygen carrying capacity as wild type IRBCs, and that's exactly what we see here. The Rh null IRBCs didn't show any any difference in oxygen binding capacity compared to the wild type IRBCs. So in summary, we've shown that gene editing and iPSC technology can be combined to generate a customized panel of rare reagent red cells for blood banks. IRBCs can undergo sufficient maturation to be used in common blood bank assays, and this has potential to be one of the first clinical applications of iPSC-derived blood cells to impact patient care. And then finally, Group O RHNL IRBCs could be used as a universal donor cell when scale-up technology is available. Finally, this project couldn't have been done with everyone listed here for the last 4 years. On top of everyone in our lab, we work really closely with the Deb and Paul's lab at CTRB, the blood bank, Malik, and people at the Muzikantov lab at Penn. We work super closely with the New York Blood Center, led by Connie Westhoff, along with our PCTC collaborators at University of Rochester and University of Toronto. So that's it, and I would be happy to take questions.